Hi and Assalamualaikum Doctor. My name is Atika binti Anwar and my matric number is 181710. Before answering the question, I will introduce a little bit about what is blockbuster drug. So, it is a drug that is typically used to treat chronic or long-term medical problems as opposed to acute or short-term conditions. Some examples of blockbuster drugs including cancer, diabetes, cholesterol, or high blood pressure medications. For the first question is how to develop the blockbuster drug. Here are simple 5S model developed by Harvard's John Quelch as a checklist of developing blockbuster drugs. So first of all is sheer size. The blockbuster drug must have an outstanding impact on a company and industry where it can break sales and records and also exceed the expectations. Second is about speed. It's not just about the sales volume, it's the speed of sales track that increases over time. Remember that the original blockbuster was a bomb that could destroy an entire city block. Blockbuster brand also should be able to fulfill customer needs very well in order to achieve and enjoy vertical sales lifts off. Third is the scarcity. The blockbuster drug should be in a very high demand where shortage and stock out occur in the market. Fourth is the sustainability. It should maintain sustainability as an excellent commercial drug that offers excellent benefits and usage to the consumers. And fifth is sizzle. A blockbuster drug does not just address an important need. It must have been done in an exciting and accessible way with excellent sales and marketing strategy. And for question number 2 is to explain the strategies to make sure that the drug can become the most potent drug. So first strategy, before I go to the strategy, so I want to explain what is potency. So potency is an expression of the activity of a drug in terms of the concentration or amount of the drug required to produce a desired effect to our body. So a highly potent drug evokes a given response at a low concentrations. Okay, first strategy is by using structure-activity relationship, which is SAR, that can predict biological activity from molecular structure by identifying its pharmacophore. Therefore, we can modify the chemical structure of the compound to enhance its effect or potency of a bioactive compound. It also is used in drug discovery to guide the acquisition or synthesis of desirable new compounds as well to further characterize existing molecules. And second strategy is the pH variation in the body. So the basic compounds is not so well absorbed in the stomach than acidic compounds. This is because acidic compounds is in an ionized form that can undergo passive absorption from gastrointestinal tract into the bloodstream. It is unfavorable for the basic compound, which is ionized or charged molecules, to pass through a lipophilic fat membrane. Third strategy is by determining the correct lipophilicity of drug. If compound is too lipophilic, it may be insoluble in aqueous media such as in gastrointestinal fluid or in blood. It also can bind too strongly to plasma proteins and therefore the free blood concentration will be too low to produce the desired effect. And also it can distribute into lipid bilayers and unable to reach inside of the cell. If the compound is too polar, it is unable to pass through the gut wall due to the decrease of the membrane solubility. And the fourth strategy is the water solubility. So the more the hydrogen bonds are, the higher the solubility of the drug. And in addition, the dissociable ions also have high solubility. Last but not least is the stereochemistry and biological activity. When the functional groups occupy proper spatial arrangement of the drug, it actually can establish a strong interaction with the receptor in the body. Thus, it can deliver its desired pharmacological effect to the body. So that's all for my answer for the two questions. So thank you, Doctor.